Welcome back student on my channel Light of Asia. So next topic, let's we starts. Eukaryotic genome organization to be continue with subtopic point number 13, nucleosome. A nucleosome is a section of DNA is wrapped around core of proteins. That this core of proteins called histone protein. Histone proteins are 8 in number, so that they are called octamer histone protein. They are representing as H1 called binding histone, H2A, H2B, H3, H4 collectively called 4 core histones. Each core histones having 2 units, it means each histone multiplied by 2 is equal to 4 multiplied by 2 is equal to 8 core histone protein, therefore called octamer. Let's come, we know more about the histone protein. Histone proteins are made up of basic amino acids. It means mostly histone protein basically made by amino acids. Basic amino acids as lysine and his family, arginine and his family. Both lysine and arginine are mostly consist in the histone proteins. It means a histone protein core having mostly lysine, arginine and his family amino acids. What is the amino acids? It is the part of the chemistry. You just study about the chemistry parts. So thank you. Let's come. Serine and his family, tyrosine and his family, and some others amino acid is also participate in the made up of the histone protein. So just we understand these 4 types of amino acids. A short discussion of structure of the nucleosome. Structure of nucleosome. A nucleosome structure having generally spherical in shape, 8 core histones unit paired in vertically. Few measurement of nucleosome structures are height of the nucleosome. It means this 55 angstrom or 5.5 nanometer. Vertically paired in vertically, it means a unit and a unit vertically arrangement. Height of length of the nucleosome, it means this length. How much? 110 angstrom, it means 11 nanometer. While the DNA is wrapped. This is the black case. Just we are structure. DNA comes here and outside here, and again wrapped here and outside here. Both the inner DNA and exit DNA, entry DNA, exit DNA, linked by H1 histone protein. Just we are this DNA, wrapped DNA size, sometimes 146 base pair, sometimes 147 base pair, but mostly 147 base pair. So just we small discussion about this. H2B, H2B, two times, it means H2B 2 units, H2B, H2B, H2B 2 units arrangement in vertically, and H2A, H2A 2 subunit arranged by vertically, and H3, H3 2 subunit arranged vertically, and H4, H4 2 subunits arranged vertically. It means 2, 2, 2, 2. 1, 2, 3, 4, 5, 6, 7, 8 octamer. This structure is octamer. H2A, H2A, H2B, H2B, and H4, H4, H3, H3, and H1 is binding, that is linker DNA, this and this linker DNA. This linker DNA having sometimes 80 base pair. It means next nucleosome, attached with this linker DNA after the 80 base pairs. What is the 80 base pairs? It means the 4 types base pair we before the discussed before lectures. You have well knowledge about the base pairs, adenine, thymine, and so on. So the pairing up is known as the base pair. Pairing of the nitrogenous bases is known as the base pair. So A, T, C, G, G, A, T, C, G, such. So after the 80 base pairs, next nucleosome occupy. Again 80 base pair, next nucleosome occupy. Such 8 unit called core histones, because it is the wrapped by DNA. It is the called core. Core, actually core word, any material occupy in the center of the any mass is known as the core. So nucleosome, it some units, it means H2B, H2A, H3, H4 only 4, and H1 linger only 5 histones. But every histone having 2 subunit, H2A, H2B. So functionally H1 protein, histone protein, actually slightly different to the others proteins. So H1 histone protein, linker histones. Actually H1 histone protein called linker histones. Why called linker histones? Actually it is the bind to linker DNA. So it is called H1 histone proteins. It is isoform. This is the matter of the chemistry, isomerism, are involved in chromatin compactation and sit at the base of the nucleosome near the DNA entry and exit. This point just we are understand and discuss. This is the entry of the DNA. This is the entry of the DNA and this is the exit of the DNA. It means that the DNA wrapped this type. It means entry and exit and entry and exit. Finally, that is finally exit this DNA and entry this DNA. Both entry and exit DNA based point H1 histone protein occupy. This is the H1. This is the H1. This is the H1. H1 bind the exit DNA and entry DNA and exit DNA. Due to the stabilize this structure, all the core histones protein stabilize due to the linker is binding. So it is binding to the linker region of the DNA. This is the linker region. This is the basic structure of nucleotide.